Okay, let's dive in. You know how ATP is basically the powerhouse molecule in every cell?
Right, the universal energy currency.
Yeah, exactly. It fuels muscle movement, nerve signals, building molecules,
Yeah,
just everything. It's absolutely foundational.
doesn't live without it.
And it's needed everywhere. Well, almost everywhere. Because for decades, there's been this really fundamental puzzle about how ATP powers one specific crucial factory inside the cell.
Ah, you're talking about the endoplasmic reticulum, the ER.
That's the one.
Yeah, it's kind of like the cell's main processing and shipping hub.
Yeah.
Huge amounts of protein folding happen there. Quality control, too, making sure everything's built right.
And lipids, right. Doesn't it make lipids?
Lipids, too, yes, and it directs traffic all over the cell. These are, well, they're absolutely vital jobs. Nonstop work.
And just thinking about those tasks, folding proteins correctly, building lipids, sorting things, you immediately know they need a massive amount of energy.
Oh, absolutely. They chew through ATP like crazy.
But here's the mystery, the really stubborn part. Scientists knew the ER needed all this energy. But for the longest time, exactly how that ATP actually got inside the ER, into that internal space.
The lumen.
Right, the lumen. How it got in there was, well, totally unclear. The door was sort of invisible.
It really was a significant blind spot, actually, in just basic cell biology. We could see the work happening. We knew it needed power.
But not how the power cable plugged in.
Precisely. The specific transporter getting the ATP into the ER across that membrane, it just wasn't identified, missing from the picture.
Well, buckle up then, because it sounds like that mystery is officially solved. Based on these sources, scientists have had a breakthrough. They've actually identified the specific protein transporter.
That's right. And the key player here is a protein called SLC35B1.
SLC35B1. Okay.
And this deep dive, well, we're going to unpack the research that figured this out, understand the how of it, and maybe most importantly, explore why this discovery could be really big news, particularly for tackling some serious human diseases.
Okay, so let's put that core finding front and center. The sources are saying SLC35B1 is the main gateway for ATP into the ER lumen.
That's the essence of it. It's the dedicated transporter people have been looking for. It solves that fundamental question.
Now, you mentioned this might not be totally out of the blue. Was this protein suspected before?
Yeah, what's interesting, and the sources point this out, is that SLC35B1 had actually been proposed as a potential ER ATP transporter before.
Oh.
But that earlier idea, it lacked the really rigorous biochemical proof. And critically, it didn't have the structural detail, the kind of molecular map needed to be, definitively accepted.
So, educated guess, maybe, but not proven.
Sort of. And there were even some other reports suggesting it might do something else entirely. So a bit controversial, perhaps.
Right, conflicting signals. So how did this team finally nail it down? What was the key evidence?
They used a really powerful approach. They collaborated with the Giulio Superti-Ferga lab and did a big genetic screen using CRISPR-Cas9.
CRISPR, okay, like gene editing.
Kind of. Think of it more like systematically switching off different genes one by one, especially genes coding for transporter proteins, to see which ones were absolutely essential for the cells to grow and survive.
Like removing different parts from a car engine to see which one stops it from running.
Exactly like that. A process of elimination, but on a massive scale. And the results were really compelling.
What did they find?
When they looked at the transporter genes that, when knocked out, severely messed up cell growth, well, SLC35B1 was right up there, among the top 5 most essential.
Top 5. Okay, that's significant.
Hugely significant. Losing this transporter really hampered the cell, which strongly suggested it was moving something incredibly vital, like ATP, into a really crucial compartment like the ER.
Okay, so that gives them the strong functional link. Yes, this protein is essential, likely for getting something critical into the ER. But how did they figure out the how? The actual mechanics? How does it physically grab ATP and move it?
Right, that's the next big step. You need to actually see the protein, see its shape, how it moves. And for that, you need a stable sample, which can be tricky with membrane proteins.
They're notoriously difficult, aren't they?
They are. So Norimichi Nomura's team did something clever. They engineered a specific antibody that targets the human SLC35B1 protein.
An antibody? Why?
It acts like a handle, or maybe scaffolding. It binds to the transporter, stabilizes it, and makes the whole complex larger and more rigid. That makes it much easier to work with for the next step.
Which is the imaging.
Exactly. It makes it suitable for cryo-electron microscopy or cryo-EM.
Cryo-EM. That amazing technique that lets scientists see the 3D structure of these tiny, tiny molecular machines, almost like taking pictures of them.
Precisely. So working with David Drew's team, using the SciLifeLab platform, they used cryo-EM to get these images of SLC35B1 bound to its antibody handle.
And what did they see? Just one static picture.
No, and this is what's really remarkable. They managed to visualize the transporter in multiple different states, different conformations.
Oh, wow. Like different steps in its process?
Yes. Imagine taking snapshots as it moves. Like a state where it's open to the outside of the ER, ready to grab an ATP molecule.
Okay.
Then maybe a closed state, and then another state where it's open to the inside, releasing the ATP into the ER lumen.
So they didn't just get a single blueprint. They got like a molecular flipbook showing the whole transport cycle.
That's a great way to put it, a flipbook.
Okay.
And that provided incredible mechanistic insight. The structures reveal exactly how SLC35B1 recognizes ATP, where it binds it.
The specific docking site.
Exactly. And the precise shape changes, the conformational shifts it undergoes to shuttle that ATP across the membrane.
Amazing detail.
And more than that, the structural map also highlighted specific amino acid residues. Those are the individual building blocks of the protein that are critical for binding the ATP and for the transport movement itself.
So, specific critical spots on the protein.
Yes, which immediately suggests potential bullseyes, really precise spots for future drug design.
Okay, this is where it gets really interesting for all of us, thinking about health. Why does knowing the exact identity and the precise mechanism of this ATP transporter matter so much beyond just understanding basic cell biology?
Well, because when things go wrong in the ER, when its function is disrupted, it's tightly linked to a whole host of really significant human diseases.
Like what kind of diseases?
We're talking about conditions like type 2 diabetes, various types of cancer, and quite a few neurodegenerative disorders, too. Alzheimer's, Parkinson's, ALS. ER problems are implicated in those.
And we often hear about ER stress and protein misfolding being involved in those conditions, right?
Absolutely. They're major culprits. If the ER isn't getting the energy it needs, the ATP, then those crucial protein folding processes can break down.
Things get backed up, proteins don't fold right.
Exactly. Misfolded proteins accumulate, the cell gets stressed, sends out alarm signals, and eventually that cellular malfunction contributes directly to disease progression.
Okay, so now, by understanding exactly how the fuel, the ATP, gets delivered via SLC35B1,
Now we have a completely new angle, a potentially much more fundamental way to think about what might be going wrong at the root of these diseases. It's about the energy supply to this critical factory.
and having that detailed molecular blueprint, the flipbook you mentioned, of SLC35B1, makes it a really concrete, tangible target for developing new therapies, doesn't it?
It really does. It's not just a vague concept of ER function anymore. We have a specific molecule, a specific mechanism.
So what's the potential strategy? Could we develop drugs that, like, tweak SLC35B1's activity?
That's the crucial question, and yes, that's the hope. Could we design molecules that modulate it? For instance, in diseases where maybe the ER is underactive, perhaps we'd want to boost ATP transport through SLC35B1.
Get more fuel.
Right. But in other situations, maybe certain cancers or inflammatory conditions, perhaps inhibiting ATP transport might be beneficial to sort of dial down an overactive or malfunctioning ER.
Interesting. So you could turn the dial up or down depending on the disease.
Potentially, yes. Targeting this specific transporter to restore energy balance within the ER could become a completely novel therapeutic approach. It's a new target, a new strategy.
And is this just theoretical, or are the researchers actually pursuing this?
No, they're actively working on it. The sources mentioned the team is already screening for small molecules, potential drug candidates that can interact with SLC35B1.
Looking for those modulators.
Exactly. Molecules that can either enhance its function or inhibit it. The explicit goal is to move towards developing these targeted therapies, finding compounds that can fine-tune the ER's energy supply based on the specific needs of a diseased cell. Well, that's a major focus now.
That's really exciting. And just for context, this foundational work we've been discussing, it was published in Nature, right?
Yes, Nature, May 21st, 2025. The title was Stepwise ATP Translocation into the Endoplasmic Reticulum by Human SLC35B1.
And it had broad support, funding from various international groups.
It did. Knut och Alice Wallenberg Stiftelse, Göran Gustafsson Foundation, AME in Japan, H2020 in Europe, JSPS in Japan. A real collaborative international effort.
Which you often see with these big fundamental discoveries. It takes a village, so to speak.
It really does. Solving a long-standing biological mystery like this often requires that kind of combined expertise and resources.
Okay, so let's try and bring this all together. Let's recap the key takeaways from this deep dive.
Sounds good.
The big mystery, decades old, of how ATP actually gets into the ER is, well, it's solved. The transporter protein, SLC35B1, is the key.
It's the gateway, and thanks to powerful techniques like cryo-EM, we now have this incredibly detailed molecular blueprint showing exactly how it works, how it recognizes, binds, and moves ATP.
And the really big, so what? Why should you care?
Well, this fundamental discovery provides a brand new target, a novel handle on a whole range of serious diseases, diabetes, cancer, neurodegenerative disorders that are all linked in some way to ER problems, to ER dysfunction.
It basically opens the door for completely new drug development strategies, right? Targeting SLC35B1 activity specifically.
Exactly. The potential to develop therapies that modulate this transporter, either boosting or dampening its activity to restore cellular health, is really quite significant.
It really makes you stop and think, doesn't it? If we can understand and potentially control the energy flow into just one specific cellular compartment, the ER, and that offers such promise for these complex diseases.
Yeah.
What other crucial cellular processes may be happening in mitochondria or the nucleus or lysosomes might we be able to understand or even correct by mapping and targeting their specific energy supply lines, their transporters?
That's a fascinating thought. Where else could precisely controlling energy transport make a difference? It suggests that understanding these fundamental transport mechanisms could be key to unlocking treatments for all sorts of conditions linked to cellular stress and malfunction. Something definitely worth pondering as this field moves forward.